With that, it gives me great pleasure to introduce our seminar speaker for today, Professor Catherine Bujold. Catherine did her bachelor's degree at McGill, which she completed in 2011. That was a bachelor's in chemistry, and followed that up with a PhD also at McGill in the group of Hannadi Slayman, where she did some really beautiful work on encapsulation of molecules within DNA nanocages and DNA-based nanotubes. She finished her PhD in 2016 and then took on a postdoctoral fellowship at Northwestern University in the group of Chad Merkin, where she continued to work with DNA and nanostructures and developed these concepts further. We were very fortunate to be able to hire Catherine in our department, in our chemistry and chemical biology department in 2020. She started in July of 2020 and had the unenviable role of trying to start up a brand new group in the middle of a pandemic with lockdowns. So, it's pretty amazing that she was able to do that. That's not easy, I'm sure. And so she has built up a group that's really interested in continuing her kind of work on oligonucleotide chemistry, DNA self-assembly, and some aspects of drug delivery. And today she's going to start a 4-part series in the BIMR lectures on DNA and sensors. And so her seminar is entitled Enhancing the Uptake and Functionality of Nucleic Acid-Based Nanostructures. And I will turn it over to Catherine to get started.
All right. Well, thank you so much for the nice introduction. I'm going to share my screen. Hopefully this works this time.
It looks good. It's just not in presenter view yet, or in presentation mode, I should say.
Yeah, so it seems my computer's a bit slow, but
it looks like it's, and now it's in the other view again.
Yeah, we'll get there. All right, this is like, you would think that after a year and a half of pandemic work, this would have been figured out, but not yet. I think now you are seeing the screen I meant for you to see, so thank you. All right, so it's my great pleasure to be here today and share some of the work I did during my postdoctoral work, which serves as a foundation for the work we'll be taking on in my group moving forward. So as you'll see, there are 2 main themes that I'm really interested in. First one is developing effective and functional DNA nanostructures, and the other one is to use them to promote cellular uptake and enhance their drug delivery abilities. So without further ado, we will start. So after a year and a half of pandemic, I think there has been one advantage to this whole situation is that many of you, if not all, are now aware that nucleic acids can act as therapeutics and that they show a very promising specificity towards addressing a whole host of diseases. And the most important example that we're all now hearing about is the COVID-19 vaccines, where there's been an mRNA that's been developed to elicit a specific immune response against the virus. So that's allowing us to be protected and hopefully to get out of this pandemic mode. With that said, nucleic acids as therapeutics offer even more promise than that. So they are probably the only drug modality that can address all levels of the central dogma of biology. So this slide really shows you how you can design nucleic acid therapeutics to really address disease that originate, for example, from genomic DNA. So I think you've probably heard about CRISPR or decoy DNA. So these can actually go into the genome and edit it directly to really get rid of genetic mutation or aberrations in gene expression. There's also probably, so that's the slightly older technology that's also extremely effective is targeting mRNA directly. So that's in your cytoplasm of your cells. You can design oligonucleotides that will hybridize with your mRNA, thus shutting down the expression of different proteins. So in this case, you can develop antisense DNA sequences or small interfering RNAs. So these are termed siRNAs, and also microRNA binding strands, which will also affect the expression of a host of genes. Finally, and that might be a little bit less familiar to you, although we have some great work happening at McMaster in other groups, which we'll see throughout this seminar, is this seminar series, not necessarily me. But really, we can design oligonucleotides to bind with proteins and surface receptors, for example. So these are called aptamers. So there's nucleic acid sequences that have a set sequence and adopt secondary structure that binds with high affinity to proteins, receptors. They can also be made to bind to small molecules in a way that's very similar to antibodies. And that can also be used to affect the progression of various diseases. So in this sense, I think what you can see is that nucleic acids, in addition to being wholly addressable, you can set the sequence to be whatever you like. You can also use them to target different aspects really of the cells, mostly nucleic acids, but also proteins and surface receptors. So it really offers quasi-infinite targeting possibilities. So with all of these great properties, it's kind of surprising that nucleic acids have not taken over the drug market yet. So there are a couple reasons for that. So if you look at linear nucleic acids, so in this slide here, I'm showing just a standard DNA or RNA strand. In its linear form, you see it's composed of three parts. You have this sugar backbone, which serves to host a different base at each repeat unit, and they're connected together with phosphate backbones. And this phosphate backbone here is usually carrying a negative charge and really defines a lot of the properties of DNA when you inject it in a living organism. So for example, DNA being negatively charged, therefore has low cellular uptake because cell membranes are also negatively charged in addition to being hydrophobic. So there's a double repelling effect here where the DNA strands really interact poorly electrostatically at the cell membrane, and they're also mismatched in terms of hydrophilicity and hydrophobicity, resulting in poor cellular uptake. That wouldn't be so bad if it wasn't for the fact that they're also promoting their own degradation by nuclease enzymes that are found in the blood or serum. And so typically injected DNA strands that we want to use for therapeutics tend to be degraded in a few minutes in your bloodstream. So that's definitely not ideal. They can also elicit immune responses. So that can be a positive or a negative depending on the application you're looking at. But obviously, that is also another factor to consider when you're developing nucleic acid therapeutics. Now, so when I started my postdoc in the American group, they had developed and put together this structure that's called a spherical nucleic acid that's found on the right. And really what it consists in is this nanoparticle core that's densely functionalized with nucleic acids on its surface in order to form like this dense shell of nucleic acids. That structure is actually really surprising because when we perform this conjugation and reorganize our nucleic acids at the nanoscales, their properties change tremendously. So while our small linear oligos didn't go into cells, the spherical nucleic acid does. And it's also able to resist nuclease degradation over much longer timescales than linear oligonucleotides will. So for this reason, these types of structures have been pushed forward for therapeutics and immunotherapeutic applications by the American group prior to my arrival. And so this here basically showcases the enhanced cellular uptake that you see with spherical nucleic acid. So if we look at this figure on the top, we have this linear oligonucleotide that's labeled with a Cy5 or cyanine 5 dye. And this is what you see as red signal in the micrographs on your right. And on the bottom, we have the same oligonucleotide, but then now templated onto a nanoparticle core to form an SNA. And what we see between these two examples is while the linear oligonucleotide does not get into cells over the 2-hour time frame from this study, the spherical nucleic acid does. So it gets detected into cells as early, sometimes as 5 minutes. In this micrograph, the first imaging was done in 30 minutes post addition of the spherical nucleic acid. And you can see a great enhancement of uptake compared to the unmodified version. So this enhanced uptake is also tremendous, and it has been demonstrated in more than 50 cell lines. So it seems to be a very general mechanism that spherical nucleic acids do get taken up by cells. However, the one key point to remember is that spherical nucleic acids are taken up via this one process that's very specific to them. So spherical nucleic acids, or SNAs, will be recognized by specific receptors on the surface of cells. These are called Scavenger Receptor-A, and some studies have shown that this is due to the ability of these receptors to detect and bind large aggregates of negative charge, which is pretty close to the definition of a spherical nucleic acid. So they are able to recognize spherical nucleic acids specifically. And upon doing so, they'll take them up into these vesicles that are called endosomes. And so once they are found in the endosome, they're kind of trapped in this compartment. And while these endosomes are very useful for promoting immune responses via the binding of TLR9 receptors, and this is something that's being explored to create cancer vaccines, they offer limited ability to the SNA to get out of this vesicle and access other cell compartments. And these cell compartments are very important if we are to target messenger RNA or genomic DNA, because the endosome ultimately typically leads to recycling of the components of the SNA outside of the cell without it being able to access the genomic DNA and mRNA in large amounts. So small amounts of the SNA do make it there, but it's still fairly small, and improving on this is a big challenge in the field. All right, so when I started my postdoc, basically the big question I was asking myself is, can we actually design SNAs that will be able to access these other compartments? I was trained in chemistry, so really my take on this was to use a chemical approach that involved modifying the nucleic acids themselves. So upon reviewing the literature, I was looking for patterns in different chemical moieties that were able to get through that cell membrane barrier, and identified guanidinium-based peptides as good candidates for doing so. So guanidinium is a functional group that's basically composed of a carbon that's bound to 3 nitrogen atoms. It carries a positive charge generally across all physiological pH. And it has this hydrogen binding pattern that enables it to bind pretty well to the fatty acids that are found on the cell membranes. And upon doing so, guanidinium functionalized peptides, or arginine-rich peptides, we should say, which arginine is the side chain or amino acid that carries the guanidinium functional group. And so these peptides are able to bind electrostatically to the cell membrane and either promote their own uptake via adaptive translocation, where the peptide is just able to cross the cell membrane on its own without the help of any active processes by the cell itself, or it will promote endocytotic mechanisms that will get it through the cell membrane as well. And so upon seeing this, I was wondering if it was possible to maybe use this motif to promote the cell entry of spherical nucleic acids. And so at this point, what we really envisaged doing was looking into ways to replace the negative charge of the phosphate backbone of our oligonucleotides with this guanidinium moiety, which would effectively now allow us to design nucleic acids that carry positive charge. And so upon reviewing the literature, we found that there were few chemistries that had been proposed for doing this. Among them, the deoxynucleic guanidine backbone that was developed by the Bruce group at UCSB, so Santa Barbara, was the one that caught our attention the most, mainly for those 3 reasons. So it retains a specific base pairing. It's synthesized from accessible building blocks. So in order to form the precursors to make this backbone, it's actually quite accessible chemistry that we could implement in our lab. And finally, the chemistry that the Bruce group put together is actually compatible with DNA synthesis. So this allows you to really incorporate this new coupling mechanism with the rest of the steps involved in the synthetic program for DNA, as well as to design and build DNA DNG chimeras, for example. So we could envisage really synthesizing oligonucleotides that would carry DNA and RNA in their natural forms and also have this deoxynucleic guanidine or DNG component added onto them. However, so that there always seems to be one negative side to all these findings, is that the access to these cationic oligonucleotides was currently limited by synthetic requirements. So I was initially quite surprised that nobody had really looked into the properties of cationic oligonucleotides in more detail, especially at the nanoscale. And it turns out that while the building blocks are quite accessible, the coupling chemistry has its ups and downs, and we're going to go over this in a little bit more detail. So first, I'm just going to give you a little bit of an overview of how DNA synthesis proceeds in case you're not familiar with it. So DNA synthesis is typically carried out using an iterative process that goes through 4 specific steps to add one nucleobase. So typically what you'll do is you'll start from a chosen nucleobase that will be functionalized onto a solid support, carrying out this dimethoxytridal protection group at the 5' end. So this group here is acid labile and easy to remove, and that reveals now a 5' hydroxyl group that is nucleophilic. So when you introduce now your coupling monomer, here it is a phosphoramidite group, which is really characterized by the presence here of this phosphorus at oxidation state 3 at the 3' position. And these are very reactive. So in the presence of an acidic activator, you can promote the rapid coupling of this phosphoramidite building block onto the growing chain through a nucleophilic attack of this hydroxyl group to form this dimerized compound here. The last step of the process now involves a capping to block off all of the hydroxyl group at the 5' position that did not react, and an oxidation to now form your phosphate linkage that will bring together your 2 DNA bases. So I went through all of this to basically show you and emphasize the importance of the phosphoramidite group here in promoting the coupling chemistry. So oftentimes when scientists want to change the DNA properties at the level of the background, at the backbone, this really involves now changing the chemistry all over to ensure that you can form new linkages and get this cycle to operate as shown here. So if you remove the phosphoramidite group, then you have to find another group that's highly reactive, that's able to promote a coupling reaction in high yields above 90%. And in few minutes, otherwise this process of synthesizing modified oligonucleotides becomes way too long and impractical to really move forward. And that's why a lot of the research in modified backbone chemistry takes some time. All right, so chemistry now. Hopefully this is something that's going to be interesting to you. So in this case, now we needed to actually replace this phosphoramidite group with another functional group that would be able to promote the fast coupling of nucleobases together and actually result in the formation of guanidinium linkages instead of the normal phosphate group that you see in DNA and RNA. And so to do this, we needed 3 different monomers. So if we look at the structure now on the left, it kind of breaks it down to you and as to which monomers would be required. So if we start here on the bottom, we have this thymidine base that was presumably attached to a solid support using standard phosphoramidite chemistry. And we can see that in order to create this guanidinium linkage here, we're going to need to have an amine at the 5' position instead of an oxygen. So in order to achieve this, that means we have to synthesize a derivatized thymidine building block that will carry a 5' amine group that's protected. So this building block here will have the phosphoramidite group at the 3' position to enable it to be attached to the solid support, but then it will switch its chemistry over to have this amine here that will now promote the formation of the guanidinium linkage. So this unit we call the initiating unit, and it begins the synthesis of these modified oligonucleotides. We then had to also synthesize a propagating unit. So this carries now, instead of the phosphoramidite group, it's a protected diurea that's used instead. So these are great precursors for the formation of guanidinium linkages through abstraction of the sulfur that's done using an activator. You can now also promote the attack of an amine at the carbon center to form your guanidinium linkage. And you can notice in the propagating unit, if you want to keep growing your oligonucleotide, you also have that 5' amine. And finally, you will need a terminating unit if you plan on doing chimeras. So that is, synthesize your cationic block with guanidinium groups and then switch over and go back to DNA. In our case, we didn't spend as much time on the terminating units. I'm not going to talk about it much today, but its main difference is that it has the 5' oxygen that allows you to go back to phosphoramidite chemistry. All right, so in terms of synthesis, so these building blocks, we synthesize them using the Bruce procedures with some modifications. In the case of the initiating unit, this was done in 5 steps. So we began with thymidine, as shown here, which we protected with a tosylate at the 5' position to make it a better Neven group during the attack of sodium azide to form a now 5' azido derivatized thymidine. This molecule was then, this as azide was then reduced using palladium over carbon to form the amine, protected with a trityl group derivative, and we then installed the phosphoramidite functional group at the 3' position for an overall yield of about 9%. We were able to confirm the synthesis of this molecule using proton NMR as well as phosphorus NMR and mass spec. For the propagating unit, it was also a similar scheme. We did that in 6 steps, also from thymidine. The main difference was that we now had to react both the 5' and the 3' position at the same time to get 2 amines in there. So in this case, we used a methane sulfonyl chloride to make 2 leaving groups at the 5' and the 3' position because it's less bulky than the tosyl group. We then performed this very interesting reaction where we boiled this molecule in base to form this bicyclic compound here, which is key in ensuring that this next attack of the sodium azide happens at the 3' position, but from the bottom, just so that we retain the natural configuration of DNA. And so that was what was done next. So we reacted with sodium azide in order to form the diazido thymidine derivative, which was reduced to form a diamino thymidine. And then we proceeded with the selective protection at the 5' position and the derivatization of the 3' position to form a thiourea using an isothiocyanate. The overall yield for this sequence was also about 9%, and the compounds were assessed by NMR and mass spec to confirm their synthesis. So once we've completed all of that, we've really had everything we needed to now start making modified backbone. So that was really exciting. And being in the Merkin group, we even had access to a DNA synthesizer to do some of this chemistry ourselves. However, when we reviewed all of the procedures for doing the coupling, it quickly became apparent that this was not going to happen. So if you look at the original reports by Bruce, the coupling chemistry to bring together this thiourea derivatized thymidine with this amino modified nucleotide on solid support, you see that the activating reagents that are required are this mercuric chloride followed by a thiophenol wash. For all of you who are even not in chemistry, you'll know it's really obvious. If you see mercury, that is not something you'll usually want to work with. Indeed, so mercury is toxic. So that was not something that was going to be easy to install on an instrument where it has a lot of fluidics and lines that need to be washed consistently between different syntheses and ensure that there were no residues for the next synthesis to proceed. Also, DNA synthesizers are usually placed on a bench in the lab, not always in a fume hood. So this was not the type of chemicals that you could really place on there and ensure that everyone's safety was guaranteed. Similarly, for those of you who have worked with thiophenol, It also is a chemical that should be absolutely maintained and kept in a fume hood or in a glove box. It definitely stinks, and it's a great way to clear a lab out of people if you feel like working on your own, but I don't recommend it. But that was basically two big pushes for us to start revisiting this coupling chemistry, because literally, I think other than a trained chemist, no one would have ever used this in a general process for making cationic oligonucleotides. And so our idea at this point was just to revisit the literature and see if there were now new methods that had been put forward to promote the formation of guanidinium linkages from thiourea building blocks using an amine to perform the nucleophilic attack. And so in this case, our readings brought us to iodine-containing reagents, so namely molecular iodine. There was Mukuyama's reagent, HTIB shown here, CMPI, so that's Sanger's reagent, and DNFB. Yes, so all of these chemicals. So the last one here is a fluorodinitrobenzene derivative. So this one did not have iodine, but all of the other ones did. And in our initial screen, what we were really surprised to see when we combined our building block that contained a thiourea and our other building block here that contained an amine, we found that molecular iodine was really effective at promoting the formation of the coupled product. All of the other reagents we used weren't so effective, but iodine surprisingly worked really well. And that led us to hypothesize that in this case, the use of an electrophilic iodine source might be the key in promoting the formation of this new guanidinium linkage. This is something we verified using N-iodosuccinimide as well as DIH. And we found that these two compounds were also effective in promoting the formation of new guanidinium linkages from these building blocks. So that was great news, because iodine is actually usually very commonly available in a lot of labs. It's also a much milder reagent. And this coupling could be performed in the presence of tetramethylpiperidine. It's a nitrogenous base that's pretty standard. And in this way, we were able to now promote the abstraction of this sulfur using iodine instead of a mercury derivative. So that was very good news for us. Another big advantage is that this reaction was actually complete within minutes. It was high yielding, and all of these reagents were easy to access. To further support our hypothesis that this coupling was promoted by an electrophilic iodine source, we were able to crystallize the iodine in the presence of tetramethylpiperidine using acetonitrile, and the crystals also with the bond lengths showed us that these were indicative of the presence of an iodine electrophilic source. This was also evidenced by this color change that we would see when we added the tetramethylpiperidine to our iodine solution. They would turn bright orange. So with this new coupling chemistry in hand, we were able to now synthesize cationic DNG oligonucleotides using a DNA synthesizer quite effectively. So we were the first to report the synthesis of a 10-mer. We had basically coupling yields that would go above 90% at every step. And this 10-mer was assessed using SDS Page as well as MALDI-TOF MS. We even made a 20-mer. I don't think I have it on my slides, but it was quite exciting to see that we could use this chemistry to synthesize now cationic oligos reliably and in much larger scales than was achieved before. So with these materials in hand, now we were finally able to start asking questions regarding the biology of these oligonucleotides, since this was a major unanswered question for the DNG backbone. It had never been synthesized in large enough quantities for cell assays. And so basically what we did is we put these oligos in a cells and synthesized a DNA strand of equal length to compare. And so in these assays, basically what we did is we incubated our 10-mer DNG with cells and compared it to its DNA equivalent. And what we saw is that in these micrographs, you see there's an evident enhancement in fluorescence signal for the DNG compared to the DNA, suggestive of a much higher uptake. And this was also further confirmed using flow cytometry, and it gave us this indication that the DNG goes in the cells in a 40-fold increase uptake compared to DNA. Another good news was that the DNG strands are non-toxic, so we were able to verify that using a toxicity assay, as well as by looking at the morphology of cells that were incubated with these strands. So to us, that was extremely exciting. And it really now set the stage for us to start exploring them at the nanoscale using spherical nucleic acids. So if you're not really familiar with nucleic acid therapeutics, these are very much highly engineered nucleic acid or oligonucleotide pieces nowadays. And oftentimes the incorporation of different charge group within the therapeutic part of the molecule is not something you want to do, as it could seriously affect their properties down the line or reduce their therapeutic effects. So what we wanted to do now is design some nanoscale carriers, such as the spherical nucleic acids for these drugs, and incorporate within them as conjugates the cationic groups that we had just been working on to see if these would enhance their cellular uptake and presumably their potency as well. So one of the questions we were wondering is, like, how many of these cationic modifications are required to have a specific biological effect, and where should we put them? Like, what's the structural arrangement that's the most efficient? So we started our studies using this SNA design, where we used a gold nanoparticle core and densely functionalized oligonucleotides onto them that were chimeras, meaning that they contained both a DNA segment and a DNG cationic part. So the DNA here is shown in blue, and the cationic part here is shown in red. So we initially started by synthesizing two different ones. So there was the unmodified 20-mer strand, and then a strand to which we incorporated 3 cationic modifications, and another one onto which we added 6. So these strands were characterized using gel electrophoresis, as you can see here on the right. And we can see from the top the number of DNG incorporations, so 0, 3, and 6. And you see that as you increase the number of positive charges, these oligonucleotides have less of a propensity to migrate on gel towards a cationic electrode. So that was the first indication of a successful synthesis, which was further confirmed using multi-mass spec. We then went about synthesizing the spherical nucleic acids themselves using these newly synthesized strands. So for this, we used 2 different approaches. The first one is salt aging, which was developed in the Merkin group and really consists in bringing together your oligonucleotides and the gold nanoparticle core and increasing the salt concentration to modulate the repulsion between the strands and the gold nanoparticle core and force them to interact with one another to form these spherical nucleic acids. We saw using the strategy that it was very effective. We had nice monodispersed bands by gel all the way to our 6 DNG modifications. We also tried the freezing directed approach that was developed by Professor Liu at Waterloo, since this method is even faster than the salt aging method. And we saw that, as they reported before, if you use unmodified nucleic acids, it works really well. However, when you increase the number of cationic charges, now you start seeing big smears and evidence of aggregation. So the freezing directed approach relies on solvent abstraction to bring together your oligos with your nanoparticle core. And in this case, it appears that the presence of cationic charge was more conducive to aggregation than the formation of well-defined conjugates. So that was an interesting finding, in that actual charge can play a role in the formation of these nanoconjugates. We also saw something interesting, is that when you incorporate the cationic charges for SNAs, you see 2 different outcomes. So the first one is that you have a reduction in overall diameter of your nanoparticles. So that's something we saw both by gel as well as by dynamic light scattering. And more importantly, at least for us, we saw that we had also a reduction in the surface charge, in the negative surface charge of these particles. And that's something we measured here using zeta potential. Now, these modification, consistent with our hypothesis, were very effective in promoting cellular uptake. So on these confocal images, you see on top is untreated, but here in the second row is T20 DNG. So that's just a normal unmodified SNA. And at the beginning of my talk, you saw how readily these go into cells. And now when we started incorporating cationic charge, it became even more evident that these were able to promote cellular uptake even further. That's shown here for 3 DNG modifications, as well as 6, where the signals really, it was really, really bright. So this was something that we were very excited about. So increasing basically the number of cationic units on the surface of your spherical nucleic acid, where they're able to engage with cell surfaces, seems to have a positive effect on cellular uptake. Even more excitingly for us is that we started seeing evidence that these spherical nucleic acids would also be able to elicit a different uptake mechanism. So this is a massive amount of bar graphs, so we'll go just through it step by step. But really, if we look at the bar graph on the left, what we did here is this study where we compared 3 spherical nucleic acids. So the first one, in like this turquoise green, is a standard unmodified spherical nucleic acid, as you would expect. And in orange, it's kind of a very similar one where we had the DNG modification placed near the surface. So it's effectively buried near the core. In red is the opposite, is we put the DNG modifications, but near the surface of the spherical nucleic acid itself. And so this is the structure we've been looking at through these slides already. So 5' means near the surface of the nanoparticle itself. So able to engage with the cell, whereas in orange, it means that these modifications are kind of effectively buried and they're near the gold core. All right, so if you look at these bar graphs now, you'll see that the orange trace and the turquoise, they're kind of similar throughout the different conditions that were tested, while the red occasionally differs. So now we're going to just go over what was actually done in this assay. So in this assay, what we did is we took cells and incubated them with different inhibitors of cellular uptake that probe different mechanisms in turn. So basically, when we look at these bar graphs, is after blocking a specific uptake mechanism, what uptake remains. And that tells us about the nanostructures being able to enter the cells using a different mechanism, or basically they're not affected by the mechanism we blocked. So let's move just through the first one. We use sodium azide and deoxyglucose to block active processes of cellular uptake, and we saw a dramatic reduction of cellular uptake of all species. However, if we look at the bars here, we see that the red one, or coral, was less affected than the other 2. Even if it's a marginal effect, like it was not as much as we expected, we still had a first indication that our SNAs, which had the positive charge near the surface and able to engage with cells, they were able to actively get in the cells, even if we blocked all active transport mechanisms. So that was interesting for us to see, however, not quite as much as we expected, to be quite honest. Similarly, we did this assay using fucoidin. So this blocks the Scavenger-A receptor that's usually able to recognize spherical nucleic acids and take them up selectively. We saw similar results once again. So dramatic decrease in cellular uptake. So what's remaining here is 20% of what we had prior to using the inhibitor. But in the case of the spherical nucleic acid with the surface modification, we saw there was more uptake remaining, consistent with its ability to, again, bypass the scavenger receptor uptake and get into the cells using different means. What was most surprising to us is when we use methyl beta cyclodextrin, which blocks lipid raft-mediated endocytosis, we had a dramatic decrease in the uptake of our DNG modified spherical nucleic acids, while the other 2 types were virtually unaffected. And that basically told us something about uptake of this specific species of DNG modified SNA, is that it's able to bind to the surface of the cell itself through mostly in the lipid raft region. And that the presence of these lipid rafts is what enables them to get taken up more effectively. So if these SNAs actually are sensitive to the composition of the cell membrane for efficient uptake, and they're able to find ways into the cells in these areas. So to us, that was extremely exciting because it was the first evidence that a backbone modified DNA or SNA was able to get into a cell using a different mechanism than the one that had been reported before. It also gave us basically the idea that we could actually control how these spherical nucleic acids get into cells if we are able to understand these processes a bit better. And so finally, we used chlorpromazine. I'm not going to spend much time on this one. As you can tell, not much happened, but this inhibitor blocks clathrin-mediated endocytosis. And in this case, we saw no specific differences between the three types of SNAs. The figure on the right really looks at this in a little bit more detail. We also incorporated in this assay SNAs that were modified with DNGs at with 6 DNGs, right? So previously we just looked at 3, now all the way up to 6, and we saw that generally we had trends that the more DNGs led to more pronounced effects. So really to us, that was now a final confirmation that backbone modifications can be used to control cellular uptake mechanisms. So really that pretty much was the end of my postdoc. That's where COVID happened. So we were not successful in completing some of the work we wanted to do on gene knockdown, but we were overall still very pleased with the outcomes that changing the chemistry of nucleic acids really changes the uptake mechanisms that are able to elicit. And that now gives us new tools for the rational design of biocompatible nanoscale nucleic acid-based nanostructures. With well-defined architecture, and it allows us to also start to determine the links between the chemical identity of the backbone modification and its properties in biological settings. And really, I think those findings were really what intrigued me the most from my postdoc and PhD work, and it's pretty much where I'm taking the group now. So a lot of the directions of our research focus on developing backbone modifications a little bit more broadly, so we can now start to interrogate cellular uptake, but using more diverse structures. So one of the chemistries we're looking at is called the phosphoramidate synthesis, and it consists in using H-phosphonate building blocks. And these are very similar to the phosphate standard normal DNA, except that the phosphate linkages here have one hydrogen instead of an oxygen at one position, which enables us to proceed with reductive amination and now incorporate different side chains on our backbone that will have different properties. So in this example here, we're just incorporating different types of amines. And what we want to investigate now is like what functional groups can be incorporated and where can we place them on DNA nanostructures such that we will have the most effective biological properties. And this is something that Joseph in my group is currently exploring using primary amine and guanidinium modifications, which he is incorporating onto SNAs. So he's successfully synthesized some of these strands and made the SNAs, and we're going to soon be looking into the biological properties of these constructs. We're also exploring bringing forth new cores for spherical nucleic acids. So in this example, it's work from Roxana and Angela in my lab. We're trying to see if we can use these modified oligonucleotides to bring in proteins into cells. And finally, we have now projects that use DNA a little bit more as a material to build now well-defined architectures. So if we think of DNA as a material, we know that it has biocompatible, it's programmable, its synthesis is addressable, and it hybridizes through specific base pairing. And when Ned Seaman pioneered this idea, in 1982, he really brought forward the first examples of well-defined hybridized DNA nanostructures that are held together with only non-covalent interactions using this precise base pairing alphabet to create 2D network and 3D nanostructures. We sorely miss him. He passed away earlier last year, but his legacy was really to bring forward the use of DNA to form well-defined architectures. In my postdoc, in my PhD in Professor Slayman's group, what she did is she took the pioneering work from Ned Seaman and incorporated to that the idea of incorporating now new functional groups onto DNA to now create new alphabets for assembly. So in this case, this is a hydrophobic polymer, and it allows us to now synthesize DNA nanostructure scaffolds, which we can now further assemble using hydrophobic interactions. And so there's lots of really interesting opportunities in this field. Now that we have nanostructure scaffolds, these are super well defined, and they're probably the most precise nanostructures that can be made to date. They also show this increased cellular uptake that was evidenced with spherical nucleic acids. So this is something that we're really excited about as a field, but they also suffer from this rapid degradation from nucleases and this low cellular uptake problem. So in our lab, what we're trying to do to address this is to try to conjugate these oligonucleotides with now cell-penetrating peptides. And this is work that Caitlin and Angela are doing. And the idea really is to try to use these peptides with specific motifs that will recognize glycosaminoglycans that are expressed on the cell surface. So in this way, that will give us now a mechanism to distinguish between different cell types and enable cell-specific cellular uptake. So this is a collaboration with the Rouleau and the Turgeon group. And finally, Kevin in my group is working now also on methods to specifically recognize different cell types using an aptamer switch that will reveal now an immune engaging motif. And this has strong applications to become an immunotherapeutic platform. So the idea is that upon recognition of the surface receptor by the aptamer, there will be a reconfiguration event that kicks out a masking motif, thus revealing an immune engaging motif that will now promote the formation of a specific immune response. And so with this, that was pretty much the overview for my group. I hope I convinced you that nucleic acid backbone modification, as well as conjugation strategies, hold a lot of promise for us to better understand how nanoscale objects interact with living systems. And by better understanding basically the impact of these modifications, as well as their placement on the structures of these nano objects, we really hope to develop now a library as well as structure activity relationships that will enable us to design better drug delivery vehicles. So that's it. With this, I'd like to thank everyone along the way. More specifically, the members of my group. So Joseph, Caitlin, Kevin, Angela, Roxana, Myra, and Rebecca. So Myra and Rebecca are actually in the Rouleau group, but they've been involved in some of this work, Angela as well. And funding sources, as well as you for your attention. So thank you so much.
Thank you very much, Catherine. That's great. I have given permission to everyone in the audience to unmute themselves and turn on their video for the question period. And so I'd like to invite people to raise their hand and ask any questions that have come up. Maybe I'll start. I really like this work. I think that these positively charged backbones are really cool that you developed in your postdoc. And I'm sure we've talked about this before, but I can't remember. Did you try to hybridize these with DNA and see if they actually base pair and form helical double helices and all this, or is that still work in progress? No,
we did. I do have the slide, but basically there are destabilizing modifications. So what we think is really happening here is the guanidinium backbone is not quite the same morphology as the phosphate. So it leads to a slightly different interbase distance. And because it's such a rigid modification, it's not able to accommodate hybridization as well as it should. So we were hoping that the electrostatic contribution would be enough to kind of mitigate that, but not quite. So it turns out, I think every base addition leads to 1 degree destabilization compared to unmodified DNA.
Okay. Yeah, I guess that's what you would expect because you're really changing the backbone, but it's interesting that they still do hybridize. That's really
Yeah.
Um,
yeah, that's this.
okay. Okay, great. Ravi, I see you have your hand up. Feel free to unmute yourself and yeah.
Yeah, perfect. So thanks, Catherine. Fantastic talk. Really enjoyed it. I don't have much of a background in DNA chemistry. I'm in mechanical engineering, but nevertheless, I was just looking at the design of this, and you chose to modify the backbone in order to reduce the charge. While from the cartoon that you had, most of the charge recognition happens at the outer end. So I was wondering, is there an end tagging that you could do to achieve the same effect as compared to a backbone modification of the DNA itself? So is there any additional value from the backbone modification that you saw?
Yes, so I think you could eventually do conjugates as well with other chemical modalities. Conjugating two nucleic acids using an enzymes would be really difficult because the modified backbone and nucleic acids are not usually substrates for most ligases anymore. So if you want to retain a molecule that's entirely formed of like nucleic acids, that conjugation strategy becomes difficult. If you accept that you want to bring in another type of molecule, such as a peptide or sugar, then sure, conjugation strategies can be readily implemented using DNA chemistry. In our case, what we were really interested in is to synthesize this molecule in one go using a synthesizer. So we would really get up in the morning, program our synthesis, install our building blocks, and be done, and have both the therapeutic part and the cellular uptake branch of it incorporated in one go, using as accessible of a chemistry and building blocks as possible, just to really limit the steps and also try to drive down costs.
Mm-hmm. Okay. The other question that I had was, by changing the backbone, do you see changes in the rigidity of the DNA itself? So, I mean, you had it at straight lines, right?
Yeah.
I'm supposing that it would be moving about and flattening a little bit. So do you see a change in that?
We don't have much direct evidence for that yet. Like, I'm going to try a little bit more placing them on nanostructures and seeing. However, measurements of persistence length, I believe, have been done mostly with AFM. I'm not super familiar, so it's very difficult for me to tell now. All I can go is that, yes, I think the rotation about the bonds to the carbon on the guanidinium linkage are probably a lot more constrained. And there's also its ability to form hydrogen bonds. And I think it forms secondary structures within itself a little bit, right? So that probably like will lead to it being slightly more rigid, but yes, I don't have direct evidence yet.
Okay, no, that's fine. Thanks once again.
Yeah, no worries.
Any other questions from the audience? Peter, please go ahead.
Yeah, thanks. So it's a very interesting talk. I don't know much about DNA either, other than what's in the pandemic. The one thing that I found interesting is your graph where you talk about the toxicity.
Yeah.
You said it's not toxic, and it had sort of the same shape as the DNA, but there was a dip in the middle.
Yeah, we had.
So is that normal? Is that known why there is a dip in that?
Yes, a slight drying effect, unfortunately. I should have repeated it, and put the newer figures in, but it was really just drying, so they all dipped together. So it's not a specific DNG effect, but rather an assay issue coming from sharing an incubator with 70 people.
Oh, I see. So that is not some effect that is...
No.
All right. Okay.
No, it was really just unfortunate. We had a hard time maintaining our moisture.
Okay, thanks.
Yeah.
Any other questions from the audience? I'll remind everyone that recordings of these talks, including today's talk, will be available on the BIMR YouTube channel probably early next week. Any other final questions? I don't see any. So I'd like everyone to join me in thanking Catherine for a really outstanding talk today, a really nice start to the 2022 seminar series, and I'll remind everyone that we will have 3 more seminars that will discuss various aspects of DNA and sensor type applications over the coming weeks this month. So thanks everyone for joining. Thank you, Catherine, once again. Have a good weekend, everyone, and I hope to see you all next week.